Hello students, I'm Dr. Girish Raisingani, Associate Professor, Department of Botany, SPC गवर्नमेंट कॉलेज अजमेर। आज हम डिस्कस करेंगे लेट ब्लाइट ऑफ पोटैटो के बारे में। द लेट ब्लाइट ऑफ potato, it attacks and kills the tops of the potato plant and invades the tubers, causing either a dry or a wet rot. It is the most serious of all the potato diseases when conditions are favorable for its development. Potato is a native of northern Andes, South America. The late blight also occurred on potato in the Andes as an endemic disease. With the development of quicker means of transport, it was almost simultaneously taken to the United States and Europe, sometimes in 1830 to 40. The trouble was well established in Ireland, England, and on the continent by 1842. By 1845, the epiphytotics of late blight became widespread throughout Europe, where it devastated the potato fields and left famine in its wake. The famous Irish famine of 1845 to 46 was due largely to the failure of the potato crop, which at that time continued the staple food of over 4 million people of Ireland. India was not invaded until between 1870 and 1880 and in 1909. Australia, which was long thought to be immune, had the disease recorded in every state. In India, the disease was first introduced into the Nilgiri Hills between 1870 and 1880. Soon after, it was reported from the Darjeeling district in the Himalayas with the introduction of English potatoes there. The physiology of the fungus did not permit it to reach the plains of India, where the temperature was relatively high for the development of the disease. During 1899 to 1900, it was observed for the first time in plains in Hooghly district of the Bengal. The disease spread throughout the state in 1901 to 2, but was again not heard from anywhere in the plains for about a decade. Severe outbreaks of the disease was reported in 1912 to 13 from Jorhat, Assam, and in 1930 from Rangpur, Bengal, and Bhagalpur, Bihar. It was reported from Pusa, Bihar in February 1928 and from Patna in February 1913. And since 1943, the late blight has been making regular appearances almost throughout the plains of northern India. However, its epiphytotics are restricted to only certain areas. This disease, it also occurs in tomatoes. Now we come on to the symptoms. At about blossoming or at any time during the growth of the plant, but mostly in January, the blight appears on the foliage, where it causes known dead spots or extended dead areas, more frequently until the leaves are killed. These blighted areas first appear as faded green patches, which soon turn to brownish black lesions, not delimited in size and enlarging rapidly in favorable weather. The dead areas appear at the tip or margin of the leaves and spread downward or inward. The rate of spread depending upon the weather. If moist weather prevails, the entire leaf may be killed in from 1 to 4 days. If dry weather follows, the appearance of lesions, the infections advances slowly and the affected areas curl and shrivel. While under moist conditions, they remain limp and soon decay, often giving an offensive smell. Very often, when the weather turns dry after appearance of the spots, the latter remain restricted in size and the dead areas look hard, easily breaking away from the rest of the lamina surface. At this stage, the symptoms are very often confused with the early blight disease by the farmers. The wet rot in warm, muggy weather advances rapidly and involves all the leaves and the stem, but it does not develop further to reach the tubers. The tubers get separate infections. Here you can see the figure. This is the late blight of potato leaf infected with late blight. It is generally the lower leaves that the first show disease, but all portions are affected, and there are numerous infections in severe cases, primary lesions appearing on petioles and stems as well as upon the leaves. When spots on individual leaflets are examined, especially when the leaves are still moist, early morning hours or after humid weather, a zone outside the purplish lesion is found to show a paler than the normal green merging into the latter, while on the lower side of the leaf, a whitish or grayish mildew growth appears on the surface of the lesion, in the surface of the lesion, and in the region where the pale and purplish tissues join. This whitish haze consists of the aerial fructifications of the parasite, which have grown out through the stomata. In dry weather, this mildew disappears. If cool, moist weather returns, the fungus becomes active and corresponding symptoms and progress reappear. In this condition, there is an abundant production of spores for rapid spread of the disease. Potato tubers are also infected while in the field and still attached to the plant, or they get infection during harvest and sometimes in the storage. And they normally, when the disease proceeds, you will find that the tuber may be rapidly it is decaying, and in storage containers also the wet rot phase of the disease is more common unless ideally cooled and dry conditions are maintained. The slimy soft decay is generally the effect of secondary bacteria which follow the late blight development. In moist atmosphere, white tufts of mycelium and sporangiophores of the fungus appear on the surface of the tuber. The causal organism. You can see the field of the potato infected by the late blight. You can see that towards right you will find the crop. It is destroyed by the disease, and on left hand side you will see the crop saved by proper spraying. The disease is caused by Phytophthora infestans. इसका जो causal organism है Phytophthora infestans. The mycelium of the fungus is endophytic, consisting of haline, much branched cenocytic hyphae, which are intercellular with single or double club-shaped haustoria or haustoroid hyphae. The sporangiophores arise from the internal mycelium through stomata and through lenticels on the tubers. They are cylinder, haline, branched and intermediate. The branching is sparse. The sporangiophores are relatively thick-walled and show cross partitions, and the side branches show the bulbous enlargements at intervals. The swellings indicate the positions where the sporangia were attached. The sporangia are multinucleate, thin-walled, haline, oval or pear-shaped. You can see in this diagram. The sporangium first develops in the tip of the branch. As soon as it matures, the tip swells slightly, proliferates, and turns the attached sporangium to the side as elongation of the sporangiophores proceed. Now the sporangium may germinate directly by means of the germ tube, and which may in turn give rise to the terminal secondary sporangium. In this figure you can see the intercellular mycelium in potato tubers with finger-like haustoria, figure 1. In figure 2, you will see sporangiophore emerging through stomata. In figure 3 and 4, sporangia with papilla, 5 to 6 differentiation of zoospores. Then 7 is escape of zoospores, and 8 to 11 is development of sporangiophores. This one. Then 12 to 15 is development of amphigynous antheridium, and 16 to 19A is paragynous, a species of Phytophthora. Now Phytophthora infestans, it comprises of several races differing in pathogenicity to host genotypes. And in India up to 1965, the physiologic race flora of the fungus was simple, only races 0, 1 and 4 being present. So now at least 14 different races of the fungus are known to occur in northeastern hills alone. So this was all about the in the first part we have discussed about the its symptoms and its causal organism. In the next lecture we will discuss about the its disease cycle and its control measures.